So that wraps up the starvation scenario. Let's turn now to scenario number 2, the well-fed scenario, which of course is driven strongly by the hormone insulin. And you can immediately see the massively broad role of insulin when we come back to our picture or our overview of the carbohydrate metabolism pathways. In effect, everything that you saw during starvation has been reversed by insulin. So in this case, we've got increased glucose uptake into insulin-responsive tissues, increased glycogenesis and storage of this glucose as glycogen, and also increased funneling of that glucose carbohydrate through glycolysis all the way down to acetyl-CoA, where it can be fed into lipogenesis to be stored as fat. So insulin plays a role in stimulating all of these steps. So let's have a look at this in more detail, starting with glucose transport. Now, we talked about the glucose transporters before, but in this scenario we've got to remember that when we're well fed, we'll have elevated levels of blood glucose concentration. So in this scenario, all of these GLUT transporters have the capacity to access glucose. But the thing to remember here is that positive association between Km and Vmax. This means that the high Km GLUT transporters, like GLUT2, will also have a really high Vmax. It's a bit like having a V8 engine that's only got fourth gear. It struggles when it's going slow at low glucose concentrations, but as soon as that speed ramps up and those glucose concentrations elevate, it really comes into its own. So of course this means that the liver can absorb a truckload of glucose when concentrations are high. Now remember that the liver is sitting on the end of the portal vein, draining your gut. So the first organ that gets to absorb this glucose is the liver. And in this way, it plays a vital buffering role for your body. It takes the top off what would otherwise be massive spikes in glucose concentration out in your systemic blood system. In effect, it offers some level of protection for your extrahepatic tissues against hyperglycemia. Now, the other key GLUT transporter that comes into its own during the well-fed state is GLUT4. And remember here, this one is responsive to insulin. You're well-fed, insulin levels will be high, and therefore a good portion of that glucose that's being absorbed will end up in your muscle and your adipose tissue, which is where this GLUT transporter is expressed strongly. Now, given the proportionate size of these two key tissue depots, muscle and fat collectively represent the other key site for glucose disposal. And so once again, the initial petitioning of glucose to target tissues is mediated through these GLUT transporters. They're absolutely crucial. It's the first thing that gets a look in. Now, a story that's got a really nice synergy with these GLUT transporters and highlights the importance of Km is associated with the very first step of glycolysis, where we phosphorylate glucose. Now, the very first thing you do after you've transported glucose into a cell is you phosphorylate it, and that locks glucose into that cell. The phosphate group acts like a big sea anchor and stops it from diffusing directly back out through that same GLUT transport channel. Now, the enzyme which catalyzes this, which is also the very first step of glycolysis, is called hexokinase. And you can see here it's got a Km of 20 micromolar. So that's a really low concentration, and therefore this enzyme has got a massively high affinity for glucose. But just remember, low Km, low Vmax. So it begs the question, what happens in the liver during the well-fed state when you've got glucose pummeling into those liver cells, diffusing through that GLUT transporter that's got a Km of between 10 and 20 millimolar? There is no way that hexokinase has got any chance of keeping up with the need to phosphorylate all that glucose. Look at its Km, it's just way too low. So your liver has got a very sneaky adaptation. It expresses another glucose phosphorylating enzyme called glucokinase. It does the same job as hexokinase, but check out its Km. In this case, it's 10 millimolar. So this means it'll also have a really high Vmax. And this makes it a way better match for the GLUT2 transporter with a Km of 10 to 20 millimolar, which has also got a really high Vmax. In effect, the two make a great synergy. They're well matched for one another. So therefore, under a high glucose intake scenario, your liver is really well equipped to be able to handle it. It's got the GLUT transporter to pull that glucose into the liver cells, and then it's also equipped with a phosphorylating enzyme, glucokinase, so that it can lock in that glucose, keeping it inside the liver cells. And while we're talking about GLUT transporters, here's the other key place where you'll find these GLUT2 transporters in action. These GLUT2 transporters are expressed in the pancreas, which interfaces with the mesenteric veins, which drain the gut. So these pancreatic GLUT2s can effectively sample the mesenteric blood, attempting to transport glucose out and into the pancreatic cells, but only if it's at a high concentration, like what you'll see after a feed. So this means that if glucose is being absorbed at a great rate, the pancreatic GLUT2s can sample it, signaling the pancreas to release insulin into the blood. And this acts almost like an early warning system, telling your body that there is a big flood of glucose coming, causing the early release of insulin, getting your muscle and your adipose tissues ready to receive that big influx of glucose. So now we move on to section 2 of our big picture, which is how glycogen metabolism changes during the well-fed state. Now, one of the central mechanisms here is the impact of insulin on phosphodiesterase. When insulin binds to its receptor, it activates this enzyme, which actually degrades cyclic AMP, turning it into 5-AMP. So in effect, this has removed that second messenger and therefore removed the stimulus of cyclic AMP-dependent protein kinase. So if you're not stimulating this enzyme anymore, it means that it's no longer trying to phosphorylate phosphorylase kinase, which in turn is no longer trying to phosphorylate and activate glycogen phosphorylase. So that's the mobilization part of the story. But on the storage part of this picture, we're also no longer trying to phosphorylate, and in this case, deactivate glycogen synthase. And of course, the other crucial thing is that we're no longer trying to activate that inhibitor protein, which would otherwise be blocking protein 1 phosphatase. So if this enzyme is active, it's going to cruise around and remove all of those phosphate groups, converting glycogen synthase to its active form, so you get glycogen storage, and converting glycogen phosphorylase and phosphorylase kinase into their inactive forms, so that you stop mobilizing glycogen. So that's fantastic. We've got reciprocal regulation, switching on glycogen storage and switching off glycogen mobilization. Now there's another even more direct effect of insulin. It activates an enzyme called insulin-sensitive protein phosphatase, which again removes that phosphate group from glycogen synthase, activating it, so that you get accelerated glycogen storage. So that's an additional, slightly more direct effect of insulin. And here's one other effect of insulin, just to add to the mix. We've already described how insulin causes GLUT4 transporters to migrate to the cell membrane so that they're actively pulling in glucose into the cell. Well, it just so happens that that glucose, once it's phosphorylated, is quite a potent stimulator of the inactive form of glycogen synthase. So that is, even when glycogen synthase is phosphorylated, if there's enough of this substrate kicking around, it'll activate that enzyme. So it's a kind of feed-forward stimulation of the inactive form of glycogen synthase. Okay, so that was the glycogen story. Now let's have a look at what happens to glycolysis and gluconeogenesis during the well-fed state. Now, of course, because we're talking about gluconeogenesis, this effect of reciprocal regulation is crucial in the liver. The most important level of control occurs at our bifunctional enzyme, PFK2 and fructose 2,6-bisphosphatase. From the previous slides, you'll recall that insulin got rid of cyclic AMP. Therefore, we're no longer trying to phosphorylate this bifunctional enzyme. In addition to that, we'll also have active protein phosphatase 1. So this will dephosphorylate our bifunctional enzyme. And this means that the PFK2 half is active and the fructose 2,6-bisphosphatase half is inactive. So we are actively making fructose 2,6-bisphosphate. And of course, that's the most potent stimulator of PFK1 and the most potent inhibitor of fructose 1,6-bisphosphatase. So in effect, we get stimulated glycolysis and we also get inhibited gluconeogenesis. Reciprocal regulation, fantastic. Now there's one additional mechanism here, a bit more linked directly to nutrition. It's through fructose 6-phosphate, which is an allosteric effector of this bifunctional enzyme molecule. So it stimulates the PFK2 half as well, producing more fructose 2,6-bisphosphatase, stimulating glycolysis and inhibiting gluconeogenesis. So in a way, that's a more direct mechanism of diet, which will increase fructose 6-phosphate, directly causing that reciprocal regulation. And the last point of control, in this case of glycolysis, occurs at the pyruvate kinase step, and this is the last step in the glycolytic pathway. Our active protein phosphatase 1 can cleave off that phosphate group, reactivating pyruvate kinase, enabling glycolysis to proceed. And so to finish this story, let's have a look at the impact of the well-fed state on pyruvate dehydrogenase activity. Now, just to get the logic right here, because we're well fed, we don't need to protect glucose. Furthermore, in the liver, when we've got an excess of glucose, we've got to convert it into fat. So it's actually quite important that this step is activated so that we can convert pyruvate into acetyl-CoA so that it can enter lipogenesis. And so therefore, in the well-fed state, it's all about switching this back on. So we need to remove that phosphate group from pyruvate dehydrogenase. And this is done by pyruvate dehydrogenase phosphatase, which, particularly in adipose tissue, is directly stimulated by insulin, returning pyruvate dehydrogenase to its active state. Now, of course, to keep pyruvate dehydrogenase active, the last thing we'd want to do is rephosphorylate it again. And therefore, we need to inhibit pyruvate dehydrogenase kinase. Now, in the well-fed state, when you've got glucose flooding into the system, you're also, as a result of that, going to end up with a lot of pyruvate floating around. And pyruvate itself is a direct inhibitor of pyruvate dehydrogenase kinase. Therefore, we keep pyruvate dehydrogenase active, allowing carbon to flux through from pyruvate into acetyl-CoA. And from there, we can either use it as energy in the TCA cycle or store it as fat. And so with that overview, you can see how when you're well fed, you increase glucose uptake into tissues, you activate glucose storage via glycogenesis, and you also allow glucose to flux through glycolysis into pyruvate, convert it into acetyl-CoA, where it can subsequently be used for energy in the TCA cycle or stored as fat.